I'm Laura Lambert. I'm an assistant professor at the University of Alabama at Birmingham, and I'm the director of our transgenic facility there as of this month. So that's exciting. And at UAB, we're doing a lot toward unraveling these variants of uncertain significance. So that'll be the focus of my talk today. We have several different centers and facilities in place for examining these, and we have a lot of really great infrastructure. So today I'll talk about the interplay of 4 of our different facilities that I'm a part of, including our neurofibromatosis program, the transgenic facility, or transgenic and genetically engineered models core facility, is our official name, and then our newest center, which is our Center for Precision Animal Modeling, that we're really excited about. And we also have a lot of interplay with the Precision Medicine Institute that helps us look at reclassifying FDA-approved therapeutics. Okay, so I'll start off just describing our facility. We're a full-service facility, so we're able to do all of the ins and outs of creating new models or rederiving models that have been previously archived, things like IVF. Today, of course, the focus of the talk is going to be our work that we do with CRISPR. We make knockouts occasionally, but the majority of our work now is in knock-ins. And then today, I'll focus on how we are making animal models of human mutant alleles, and the majority of which are SNPs, but occasionally we'll get a partial humanization or some larger change, but for the most part, these projects will be SNPs that I'll show you. So for anyone not familiar with how we make rodent models, this is just a quick workflow of what we do. So we take our CRISPR reagents, all of them ordered through Synthigo, and we inject them as ribonucleoproteins directly into the pronucleus of a fertilized embryo. We transfer those into what you can think of as a surrogate mother to carry them to term. And then once the pups are born, we extract genomic DNA from the tails, and then we genotype them. And we have several different assays, but our workhorse that you'll see throughout the presentation is really just PCR and then running them out on a polyacrylamide gel to get really fine resolution and look for any CRISPR-induced changes. And then, of course, we confirm everything through sequencing. Okay, so like I said, at UAB, we are really a hub for neurofibromatosis research, and a lot of our work is in neurofibromatosis type 1. The disease itself won't be the focus of the talk today, but I'd like to think of it more as like a platform for the types of modeling that we're able to do, particularly because it is a rare disease, and that's the focus of a lot of my work. So we do a lot of genetic testing for NF1 at UAB, and many times with a genetic disease like this, we come across variants that we're unsure of the impact that they have. So, variants of uncertain significance. And the question is, how can we prove whether or not this is a causative variant? And so we take a lot of these different variants and we look through them. We look for similarities and differences and try to, we can't model everything. So we try to make really intentional decisions about where we want to model them, look at different domains of the protein, and so on. One that was really interesting and stuck out is this mutation that leads to a clinical subset of NF1 called familial spinal NF1, where the patients develop tumors down the spinal cord. It's distinct in presentation from traditional NF1. And at UAB, we do have the ability to make rat models in addition to mouse models. So we set out to replicate this particular patient mutation in the rat, and we got the most striking phenotype. This is a spontaneous breast cancer model in the heterozygous state. So this animal has one copy of the patient allele, this R1276Q mutation. The other allele is initially wild type, although we found that in the tumors, there has been a loss of heterozygosity. And what was exciting for us to see was that this model seems to be recapitulating the pleiotropy that is seen in the human patient. So you can see when we dissect out the spinal columns, if the animals are able to age out with this enormous tumor burden, We do get some animals that have nodules along the spine or pheochromocytomas, which is another phenotype seen in NF1 patients. And then you can see over here just how enormous these tumors can get. Okay, so as we're going through this process of creating animal models of these rare variants, we end up with this entire suite of NF1 rodent models that there for us to use, and we openly and willingly share them with other groups. But what's been really interesting is that now that we've built this suite of animal models, we are not just looking at these models as individuals anymore, but we can look at them more as classes for treatments with therapeutics, and that's exactly what's been done here. So you can see all of the different research initiatives that we have going on at UAB, targeting them more on the basis of the effect that they have on the RNA or protein level. So we have an investigator looking at exon skipping for some of the splice variants, and then or ribozyme editing, nonsense suppression in the case of our stop mutations. And then you can see the different types of approaches that we've had to take. So making these patient variants, as many of you know, you don't have a lot of room for variation in where you can target and the way that you can target. And so we've had to get a little bit creative with some of our approaches. We've had to use alternatives to Cas9 like CPF1 or also known as Cas12a. We've done electroporations. In the case of downstream therapeutic approaches like exon skipping, we really need that entire exon to be humanized, because the antisense oligos are going to bind directly to that sequence, and we can't optimize them binding against mouse. So have to make bigger changes in that case. One of the initiatives I'll take a moment to highlight is the research of my long-term mentor, Dr. Robert Kesterson, who's doing nanoparticle gene therapy, and he is using the rat that I just described to you that has that really striking breast cancer phenotype. And so we used CRISPR to make the rat, and now the idea is that we're going to use CRISPR in nanoparticles to go back as a gene therapy and edit that mutant allele back to wild type. So we're fortunate that there's a guide that binds specifically to the mutant allele, and it does not recognize wild type. So once we encapsulate that, we can deliver it directly into the mammary glands and see if we can convert enough of the mutant allele back to wild type that we could have some positive effect on the phenotype. And our first dose with the nanoparticle treatments is actually using mouse wild type cDNA just to test delivery and see what types of levels of expression that we can get. But you can see here, once we encapsulate that wild type mouse cDNA plasmid along with an RFP in the nanoparticles and deliver them, we went in and dissected out the mammary glands, and we're getting really nice expression. So we know we're in the right place. We know we're getting delivery. We know it's expressing. So that's really exciting. Okay, so I'll switch gears just a little bit to focus on CPAM now, although I hope by the end it all ties in together and you can see how nicely these groups flow back and forth. But this is our newly funded center, CPAM for short, Center for Precision Animal Modeling. And this is really, to me, this is the culmination of all the work that we've done at UAB in modeling not just rare diseases, but any types of patient alleles. So just, we build up this repertoire and this reputation for being able to do it. And then we're able to package that all together, not just in rodents, but with multiple model systems, and become one of 3 centers in the United States that's funded to do this type of work. So there are 5 different units in CPAM, and so you can see how we all go back and forth. Here in the middle is the RSS for short. But what's important to note here is that they're technically 5 distinct units, but we all work extremely collaboratively and are all kind of involved across the board as much as we can be. Okay, so we have 4 different in vivo systems that could potentially be used to model any nominated variant that makes it through the pipeline. So we have worms, fish, mice, and rats. We in addition to that, also have a cDNA screen that we can do in Xenopus. And we also have an in vitro system, so we can, of course, take any of these patient mutations and put them into a cell culture system and test therapeutics that way as well. Our timelines here, so, there are differences in how long it takes to make different species and how labor intensive it is and their generation times and so on. So we have our timeline here of when you could expect to see a worm or a fish or a mouse version of the mutation that you're trying to make. And we had to keep this in mind when we were proposing how many different models we could take on per year and what our capacity would be, because it is a lot more expensive and time consuming to make a rat, for example, versus a worm. And I want to take a moment to really emphasize that we are eagerly soliciting external nominations. So this is not a closed system, or this is not a UAB only system. We have a landing page on any browser. There's actually a web form you can access through your cell phone where a physician can nominate a variant for consideration, and really any variant that is nominated, we will take a look at it and we'll see if it's a good fit for our program. So we really go through them with a fine-tooth comb and make sure that it's something that we can readily take on and model, and something that we can also target therapeutically downstream. In the interest of time, I'm going to breeze through this slide a little bit. I just wanted to show where our nominations come from and emphasize again, we do solicit external nominations, as well as the nominations we get through the Precision Medicine Institute or the Undiagnosed Diseases Program and the CCTS network. And so as these come through our workflow, the idea is that we'll be able to get back to the patient at some level and have some type of effect on their clinical care. That's our ultimate goal. So this is our workflow of how we determine if we're going to make a model, and then perhaps, if we're going to make a model, which one is going to be the best. And we really have it's some intense conversations and meetings. So every Monday at 4 we meet and we review these cases, and the fish people will say, I really think this should be a fish, or us rodent people will say, no, it should be a mouse, or we may end up saying that it should be both, and we'll do both of them as long as we have the resources available to do it. And I'll share these slides later so you guys can take a closer look at them. Okay, so hopefully bringing this all together is our NF1 CPAM variant that was approved. Okay, so of course, as we're getting our feet under us with CPAM, we needed to have some demonstration projects. And so we really capitalized on the expertise of existing UAB investigators who maybe had some other types of animal models in that field, or had a rare patient variant in mind that they'd been wanting to work with. And so, of course, one of our overarching themes was RASopathies, which encompasses NF1. And so this same variant that we modeled in the rat, of course, one of the questions I frequently get when I present on this animal model is, does it do the same thing in the mouse? Is this species specific, or is it variant specific? And so to get to the bottom of that, we nominated the variant for CPAM, and as it went through the full workup and workflow as any other variant, it was accepted for us to make a mouse and actually to make a fish of that same variant. So we'll end up with a suite of animal models. And excitingly, we've had some early success in generating these. So this is a mouse. This is the mouse model version of the rat that I showed you earlier. And this is the early CRISPR targeting we're doing for that. So we've got our frequencies down here. We were absolutely delighted to see that of 12 pups born, one of them had the knock-in. And then we went through and sequence confirmed that. So our systems are up and running. We're getting the sequence confirmed founders and rodents, and everything seems to be going well. So to give an overview of the number and types of projects that we can take on and what our capacity is, so far 65 projects have been submitted. We're looking for, we have more that we're going to be reviewing, but we're also looking forward to the coming years taking on more and allocating them out because we have the resources to take on this kind of work. And in addition to creating the animal models, we have an entire unit dedicated to sharing them. So we're going to be depositing them to national databases or sending them out to anyone who wants them. So a lot of work to do. Hopefully, I have shown you how we're having an impact on clinical care through studying rare disease variants at UAB through all of our different units that work together. And I'll take a quick moment to acknowledge everyone in my lab. And also take a second just to say that we do have a microinjectionist position open. So if you have any expertise in that area, please contact me and I'd love to talk to you. So I don't know if we have time, but I will take your questions if we do. Okay. Can I provide gene editing services internationally? So we certainly can do that through TGEMS. CPAM is an NIH-funded center, so that would be restricted, but we have made plenty of models through TGEMS for international institutes, particularly in India so far, but that's not an issue. I mean, there's a lot of legalese to get through, but we're able to do it, sure.